I'd like to introduce the next speaker. His name is Piotr Kosla. He's dialing from Poland. So he's a proud father of 3.5 years old twin girls, Lena and Zuzia. Piotr comes from Poland and where he currently lives with his family. His professional background is finance and international management. He graduated from Warsaw School of Economics and National University of Singapore. Before joining Biogen, he spent several years in finance and business development in several big firms. 3 years ago, Piotr's daughter Lena was diagnosed with the ultra-rare genetic disorder, PACS2 syndrome, PAX2 syndrome. He and his wife founded the PAX2 Research Foundation, driving treatment discovery for about 150 people affected by this disorder. In 2022, he reached out to us for iPSC generation for his daughter without knowing too much background of this disease. And ever since then, I've been following his journey in developing the treatment for children with PAX2 condition and was super impressed by what he has achieved with only 3 years. And today we are going to share he's going to share his journey with us. Peter, can you dial in?
Yep. Hello, everyone. First of all, can you hear me and can you see me? That's the most important.
We can hear you. We're working on a slideshow.
Okay, perfect. So let me first. Okay, I'm trying to share my screen. Just wait a sec.
Yes.
So as long as you share the screen, I cannot share my screen. So you have to stop sharing the screen.
Yes, hi, Piotr. I just stopped sharing.
Almost there.
Perfect. Okay.
Let's see how it goes.
Perfect. We can see that loud and clear.
Okay, that's great. That's great. Thank you. So first of all, welcome everyone. Good morning to everyone. I guess it's around exactly 9 a.m. On my side, it's 6 p.m. So I'm dialing in, as it was mentioned, all the way from Poland, which is 1,000 miles away. But it doesn't stop us from collaborating together with iXCells and with many other parties, which I'm going to mention during my presentation. But before I get there, I wanted to say a big thank you to Michelle for having a great and very emotional presentation. And she always inspired me as well in my job around the PAX2 when I see what she's achieving and what steps she's making with KCNH1. So thank you, Michelle, for that. And I hope at some point we'll get to meet each other in person. Because for those who don't know, we've met like 2 years ago. And every now and then we update each other, how we're doing. And I'm always inspired by the amount of motivation that Michelle is having to push the treatment development further for all the kids with the KCNH1. So thank you for that. Moving to our case. So today I want to talk for, let's say, 15-20 minutes about the journey we did in the last 3 years when we were starting completely, and I mean it by saying completely from scratch, when there was like no interest around the PAX2 syndrome at all back in 2020-22, and where we are today after 2.5 years, to be very specific, from the moment we decided to do something about it. So as it was mentioned, me together with my wife, so my name is Piotr Kosla, and with my lovely wife on the left-hand side, Małgorzata, Gosia Kosla, we do have some background, which in fact helped us to, let's say, start a bit faster than some of the other parents that decide to take care of developing the drug for the rare diseases, because I have some medical background, as well as my wife. However, both of us, we finished business schools. We didn't study medicine. We didn't study biotechnology and any of that. So we had to learn very, very fast. But something I'm gonna link during the presentation. I'm going to link the fact that the fact that we finished the business school helped us a little bit at the very beginning to, let's say, to manage a project. Because at the end of the day, I guess every drug development is a project. That also requires specific competences to push it further. And on the right-hand side, you can see our reason. So Lena, that today she's 3.5 years old, but she has twin sister, which is very important here, also from the research perspective. They are fraternal, as you can see also from the picture. But in fact, it allowed us to move quite fast with the cell painting research, which I'm going to talk about later on, and how Zuzia cells were beneficial in advancing the treatment development for PAX2. But starting from the very beginning, so basically our story. So before December 2021, we're a normal family with with two kids, with twins, which were back then 3 months old, and we didn't see it coming that at some point Lena will have the first epilepsy, which very quickly moved into the status epilepticus because of the fact that they were twins. Some of like Lena, she had some issues with the tension in the body, together with Zuzia, but it was always explained by the fact that they are twins. So as simple as that. So, and apart from that, everything was fine. So if you look at Lena when she was 1 month old, 2 months old, 3 months old, you wouldn't tell that she might be having some devastating disease in front of her. But in December 2021, during the holiday time, she experienced her first epilepsy, which followed by 30 plus in 48 hours when she was in the status epilepticus in the hospital. And then quite quickly, we've managed to diagnose her thanks to whole exome sequencing in February 2022 during Valentine's Day. So it was exactly 3 years ago, as we have 18th of February today. And we quickly learned that she's going to face a very rough and unpleasant future because of the clinical phenotype that was described in the first publication from Heather Olson. And when you read the publication, you see that she will be facing intellectual disability, autism spectrum, psychomotor delay, cerebral dysgenesis, and so on. So very similar to other DEEs as well, and also to what Michelle was speaking about like minutes ago. And back then, Lena was second in Poland, and around 40 case diagnosed globally. So as you can imagine, the interest around the disease was very limited. Back then, when we decided to start the foundation in May 2022, there was 0 scientific publications on the disease, apart from the fact that there were some case reports, and only one scientist, one professor interested in the PAX2 syndrome, and only because he was studying healthy gene. So at some point, he also decided to spend some time understanding the syndrome itself, and that was it. So that was our starting point. So really from scratch. And then I will tell you, how we, what kind of, what kind of progress we made in May 2023, May 2024, and where we are today in our journey. And at the very beginning, that's all we knew about PAX2. So we knew what is the healthy gene role, what is the protein structure, and as you can see, the PAX2 is located in a highly unstructured region, and that there were like cases of mutations in E21IK and E11K, which were leading to the disease. And the last thing was that the PAX2 gene expression was highly expressed in brain and skeletal muscles. And regarding brain, it was mainly oligodendrocytes, and that was it. So this is what we started from, and also from case reports, which were first described by Heather from Boston Children's Hospital, explaining how the clinical phenotype looks like. But today, before I move to the scientific part, today we are connected to more than 150 patients worldwide. And as you can see, not only we are connected to the patients, but we are also connected to the physicians, because building on what Michelle was saying about the natural history study, we are also preparing for that, because we are trying to tackle everything at the same time. So not only focus on the research itself, but also looking from the clinical perspective, what will be needed in case treatment is ready. However, what we see is that the same mutation variant, because 95% of the cases of PAX2 are kids with E21IK mutation, really like 95 or even 98% of the cases. However, phenotype differs a lot. So we have different onset of epilepsy. We have different spectrum of intellectual disability from mild to severe, or even we start to see patients with borderline or even almost normal intellectual development. You can have autism spectrum disorder. Most of the kids, they have it. And the same with verbality and non-verbality. More than 3/4 of the kids are non-verbal. So, but the striking thing is that despite the same variant, you have a really very, very phenotype of the disease. And we also try to understand why is that happening. And in May 2022, when we decided to start the whole journey with the PAX2, this is where we were in terms of the reagents. So it was quite lucky that we found out very quickly that very close to Poland, in Czech Republic, today called Czechia, there was actually non-humanized mouse model which was frozen. And we found about it by coincidence in a way. And that was our kind of situation that, hey, so we kind of have potentially non-humanized mouse model with the PAX2, which is frozen. Let's do something about it. And then this is where we are today. So down the line, when we decided to start the whole journey, we got into collaboration with a couple of different places all around the world, including iXCells, because as Niawei mentioned, in fact, the fact that we are here in Poland and it didn't stop us from sending the blood all the way via FedEx from Warsaw to San Diego. And in fact, the first iPSC line was generated with iXCells, which was then in person delivered by Niawei to Northern Foundation, which I'm going to talk about in a second. But today we are basically we have I mean maybe not all, but most important reagents to work on, meaning fibroblasts, iPSC lines, neuronal cells, both humanized and non-humanized mouse model. And with some of them, like for example, you can see here with the non-humanized mouse model, we actually did some initial phenotyping as well. So we are kind of try to move as fast as possible. But what is very crucial is that from the very beginning, we have this approach of doing everything at the same time. So we focus on basic research, translational research, clinics, all at the same time. And from our perspective, it's all about the cooperation. It's all about reducing the bottlenecks from administrative perspective, and also maximize the investment. So we are looking at every dollar from every side. And what we also do, we try to monitor all the work on the PAX2 syndrome across the globe. So in case there's something new happening around the PAX2, very often researchers, scientists, they approach us first, and we straight away we update them what is happening in all the places in the whole world, so that we don't duplicate the job, and we allow this flow of information between everyone involved in our journey. So as you can see here on the slide, we are focusing on the in vitro, in vivo aspect. From the translational research, we are super lucky to be collaborating with the NLOREM, because Lena is part of the NLOREM family since late 2022, and that's the most promising approach in PAX2 syndrome. Silencing the mutated gene, or the whole gene as such. But we are also trying to approach the problem through drug repurposing, and I'm going to talk about it in a second. But we are also looking at more long-term perspective. I'm talking 5+ years from now, regarding RNA editing, genome editing in mice. And last but not least, as I mentioned, we are focusing on collaborating intercontinentally on the natural history study, because with such limited patient pool, sometimes it's sometimes it's impossible to do the proper natural history study within one country because of the number of the patients. Because obviously doing the natural history study with 2, 4, 5, 10 patients is not enough. So that's why from the very beginning we work intercontinentally, as you can see here, for example, between Boston Children's and our medical university here in Warsaw. And we are part of many different initiatives like EpiCare, which is epilepsy care in Europe, or Simon Searchlight. Today we do have some progress around PAX2 on many fronts. So first of all, we have a couple of compounds as a potential drug repurposing candidates based on our cell painting study research with Charles River. As I mentioned already, but I'm gonna mention it once again. Lena is part of NLOREM family, so we are part of this active program for PAX2 syndrome. Hand in hand with many different diseases, including KCNH1, as Michelle mentioned. We do have the mouse model ready for the preclinical examination, but we are also focusing on things like publications. We do know it's important also for the researchers to publish about their job, and we try to do anything we can to support it. And we are also focusing at some point to move from crowdfunding and using our private funds, because something that I didn't mention is that 95% of things we're doing is our private money, either our private money or crowdfunded through friends and friends of friends and so on. However, we are doing also some progress around the grants. So we got 2 grants awarded for basic research in 2024 from NIH. So we're actually helping or closely collaborating with researchers that actually got them from Pittsburgh and from Chicago. However, we are not stopping here. We are in process of submitting or awaiting decisions for different grants all around the world, including US and Europe. So kind of to summarize, before I move to the video, kind of summarize the situation right now. So from May 2022, with 1 interest among scientists and 1 project of the Paxton syndrome, today we have more than 50 scientists which are somehow involved in the disease, and more than 10 active projects happening all across the world. Which I wanted to kind of highlight here is that it doesn't matter that we started from 2 patients in Poland and 40 patients in the whole world. I tend to say that today more scientists are involved in the project than number of patients diagnosed 3 years ago, which for me is amazing to be able to say that. And also to kind of to send the message out there is that, you know, at the end of the day, science doesn't choose, and science doesn't differentiate if you have 2,000 or 2 million patients. You know, it's all about the science at the end of the day, and you can make some scientific breakthrough even when you decide to spend the time on such a ultra rare disease like Paxton syndrome. And at some point, we decided to make things more official and kind of, you know, collaborate even closer with the community. So as I mentioned, we do have a couple of families which we are connected to, but also a couple of scientists which we are connected to. And at some point, we decided to kind of, you know, bring them as much as we could bring them together to Poland. So actually, we had a couple of people flying all the way from the US, from UK, from the Netherlands back in September last year for our first conference, which was mainly focused around the Paxton syndrome, but we're also talking overall about the rare diseases and what is happening, you know, in both US and Europe and all across the globe around the rare diseases as such. And before I move to the last slide around the advocacy that we are doing in Poland and in Europe, let me invite you for a small video, and hopefully it will be working and the audio will be there.
So welcome on the conference.
I'm just amazed by how many strides have been made in such a short time and how many people have been activated to the cause.
The scientific level was very, very high. I got to interact and meet a lot of very interesting people. I think I come back home with a very renovated vigor, let's say, to work on PAX2 on a lot of rare disease challenges.
That also allows me to see the patients, their problems, their disorder or their disorders in different ways that would allow for different approaches and different treatments.
I'm surprised. I'm surprised because I find that it's extremely incredible how you manage to bring, even though it is a ultra-rare disease, small community, that you manage to bring so diverse people with such a different experience, with such an excellent scientific talks, with people, you know, with really from the diverse horizons.
So I hope the audio and the video is working. And the last thing I wanted to tell you is that, you know, when you focus on the rare disease, when you focus on the ultra-rare disease, very often, naturally, you start to advocate for the whole community. So obviously, 80% of our time we are spending on PAX2, but every month it's moving towards, the percentage is growing towards overall rare diseases. And we are advocating around it in Poland, we are advocating around it in Europe and also in the US. For example, here on the left-hand side, you can read that we were invited to open the Symposium of UK platform of nucleic acid therapies as the main family speaking around the progress we have done. On the right-hand side, on the top, you can see us talking with actually the Polish Minister of Health, Izabela Leszczyna, about the need for the increased public support for the innovative projects on the rare disease therapies. And last but not least, what is for me very proud is, despite the fact, again, we are talking about the disease where we have today 7 patients in Poland and roughly 150 to 200 diagnosed globally. So you could say, hey, we are talking about a very small community in Poland, but despite this fact, we were noticed by the Ministry of Health and Polish government. And my wife, she was invited to join the Council for the Rare Disease at the Polish Ministry of Health, which is very prestigious council because there are only 3 patient representation there and around 50 to 20 most important professors which are involved, which are actually day-to-day involved in the rare diseases, together with the representatives of the Polish government, which allows us also to advocate for the overall community and access to innovative therapies and innovative projects, because it's not only about therapies, it's also about allowing a regulatory pathway to be able to treat these patients faster. Because in some of the situations, we actually sometimes have the treatment, but if you don't have the regulatory pathway that allows you to move fast, you tend to lose time with the treatment being there on the shelf. So this is also what we are doing. We are advocating for overall the community, which also is important right now in Poland because we are leading the European presidency, which also is allowing us to to target and focus on more and more people, not only in Poland but in the whole European Union and Europe as such. And that was it. So thank you for having me here. And again, I'm super happy that I was able to talk once again on the iXCells Day around the rare diseases, because I was able to talk about it 2 years ago. And once again, I want to say that despite the fact that we are miles and miles away from each other, it didn't stop us from collaborating together. It didn't stop us, the parents, from jumping on the plane and flying all the way from Warsaw, for example, to Boston for the NRM meeting every year, or meeting other scientists in a different part of the world. So thank you for that, and I leave it to the last presenter from patient's perspective.